we will speak about the way how we can get to that point where we will have trees. So for creating trees, phylogenetic trees or genetic distance trees or time trees, what you need is a distance matrix. A distance matrix is just a n times n table. It's like an Excel table. If you have 100 isolates, you will have a 100 times 100. So 100 rows and 100 column table where you have a distance value between each 100 isolates or species one by one. So it's a pairwise distance always. You have the distance between number 1 and number 5 or 26 and 31. And this is the distance matrix which will be used by any other algorithms, clusterization algorithms, clustering algorithms to make trees. And I think we will speak about different algorithms on Thursday to produce the tree from this. But today we will I hope we will get to that point where we will have some kind of a tree from, for example, a few isolates. So the reason I sent you a few links, and I would like to discuss different methods here, because the first thing you have to decide when you start creating your genetic distances is the approach. You have to know, at least in some way, what you are comparing. If you are comparing the same species but different isolates, or if you are comparing exactly the same strain but in different time points. For example, if you are thinking about a mutation accumulation experiment in many of your projects, then you can be sure that the genome is extremely similar. So you don't really have to think about what kind of extra genes I have here or there, or is there any overlap that I can use to do a multiple alignment. So the first thing you have to understand or decide how to get to the alignment phase, if you can do an alignment. So that's one of the main phases of genetic distance calculation, that you have a pairwise or a multiple sequence alignment. As soon as you have one gene, or you have multiple genes, or maybe you have a whole genome that you can align to each other, then you can just simply count the differences. And again, actually, the final number in that n times n table will be some kind of number that is reflecting to your own calculation, or the calculation you are doing with the program. So let's say that you have you are working with 1,000 nucleotide windows, and you are comparing whole genomes. And between two genomes, you find, on average, 5 or 10 nucleotide differences. Let's say that we are having 10 nucleotide differences on average within every 1,000 nucleotide window. So that means that in terms of similarity or distance, they are 99.9% similar. So you can use this number, or you can use the 1 minus 0.999. It depends on the distance matrix you use. It depends on the program you will use later on. You can have a similarity, or you can have a distance matrix. In similarity matrices, 1 means that they are completely similar, and 0 means, of course, that they are completely different. In distance matrices, 1 means that they are completely different and they are the most further from each other, and 0 means that they are zero distance from each other. This is the reason why I tell you this, because there are different outputs from different programs. Sometimes you get back a distance matrix and sometimes you get back a similarity matrix. But as soon as you know what you have and you know what you need for your next step, it's very easy just to convert them. So basically the conversion between a similarity and a distance matrix is just simply 1 minus x. So for all the numbers within your table, subtract the number from 1, and you are just converting it to the other type of matrix. So the first question is, when you are trying to make this phylogenetic or genetic distance tree, let's just first speak about genetic distance tree. Don't really use the phylogenetic word here. The genetic distance tree, if you go for an alignment-based one, because you are pretty sure that there will be genetic regions that are ready to align, then, okay, let's go from the most similar ones to the less similar ones. If you are working with 10 or 20 different isolates from the same, for example, mutation accumulation experiment, then you are pretty sure that you can align the whole genomes to each other. That means that the best way to do the alignment is to do a whole genome alignment and whole genome SNP alignment, and this will be done by SNP. Yeah, I will just close this window, and I'm not here, but I have this one here. No, so we already used SNP before. Today I will speak about a lot of theory, and forgive me that because of this, because we just simply don't have time to run all of these what I want to show you. I already promised you to make a cheat sheet from realignment, which I haven't done yet, but I will do this first because I think this is much more important for you, for all of you. So let me just do a step-by-step guide how you can use these. And today, please just try to understand and how you have to start doing what you do, or start doing these kind of different approaches. So if you remember, we used SNP for looking at the difference of one isolate row sequence data compared to a known reference sequence. But if you go down with SNP, you can also do it for multiple isolates. That's what you can do with the SNP multi. It's actually the same thing what you would do with a single isolate. So SNP multi is actually running multiple SNP processes, and at the end, you will be able to, sorry, use the final output, which is here, to gain the tree or the gain the distance matrix. So you just simply have to follow these steps, which again, I don't want to go through now. I just want to tell you when you have to use this. So again, as I told you before, SNP is very good if you have a resequencing project. If you are sequencing something that you already know very well, and this is why here, especially with the prokaryotic word, this is a huge trap. You shouldn't use SNP alone if your isolates or if your cultured bacteria are not very close to each other. So I hope that Chioma don't mind, but let's just have Chioma's example when she's collecting a lot of different salmonella isolates from Nigeria. as soon as you are collecting different samples from different geographical locations, you cannot be absolutely sure that what you are sequencing are completely the same genomically. There is a lot of chance to have different plasmids or even different gene sets within the genome between these isolates. If you are using a single reference genome, which you have to use for SNP, then you will only find out the differences compared to that reference genome. So you can use, of course, a known salmonella reference genome. You can align all of your isolates, but the only thing you will find out is how the aligning parts of your genomes are different. It's definitely not good for finding new genes or finding accessory genome differences. I hope this is clear. If anything is a bit messy when I'm saying, just please stop me or ask me on to stop on chat, especially if I will open the chat window as well. But there are also very good things with SNP. So SNP is always producing the VCF file, as we saw before. So if you, for example, again, going back to the mutation accumulation project, if you are looking for those mutations that were happening, and especially for mutations that have some kind of effect on protein level, then SNP is a very useful tool. You can also follow in very high detail the mutation rate. That's also extremely good with the whole genome SNP alignment. So you can really define a mutation rate, X number of SNPs per Y number of replications. And also, if you are thinking about those projects, for example, when you try to make a time tree, again, from the same species, from the same strain usually, then I would say that one of the best approach is to do a whole genome SNP alignment. But again, it is good for timing your tree. It is good for finding alterations within genes, but it's not good for finding novel genes, novel gene sets, or accessory genomes. So that's why we are going to the next category, which is the core genome-based alignment, and then together with the core genome-based alignment, looking for the accessory genome. That's what I would think about using Rory, and what I used Rory for a very long time, but according to Julian, we should use Panaru. So I installed Panaru for myself. And again, I will give you a step-by-step guide as well, again, how to use it. The problem with Panaru, so there is a documentation, as you can see on my, so if you are looking for the GitHub, and that was why my problem first, usually if you have any programs on the GitHub, you just go to the README file, or you just go to the docs and then the README file, which is here, and then you know how to use it, how to install. This is usually what you get back when you, I don't want to compare Panaru to Proca, but or Rory, but this time I have to. So when you just open a GitHub page, usually what you see here is a lot of information about the program, how to use, how to install. This is what we did before with many of you when we installed Proca, Rory, or anything else. And this is actually within the readme.md file, usually. This is the standard that you are putting your readme.md file in the root of your repository, and that will contain everything about installation and usage. In Panaru, this is not really what we have. And the readme.md file is only containing this link, which is also good. So we will have a documentation somewhere else, and I will show you soon. But if you, for example, you want to just simply download or clone this repository and you are looking for the documentations which is inside your download folder, then you will find this, which is, I think, outdated probably. This is probably one of the first versions of Panaru. If you try to run Panaru like this, it won't run. But even this one will not run. So if you just look at the Panaru, you don't have it yet, so just please look at my screen. And -h, which is the help. And then you can see that there is one obligatory mode option, which is --clean -mode. According to the documentation, it's --mode. I haven't tried with --mode, but I just don't like when the option, the real option, is different from the option which is on the documentation. And I think the main difference between Panaru and Rory is here. So the strict mode is, I think, very comparable to Rory, but there are moderate and sensitive modes as well, when there will be a bit less strict comparison between the strains you are putting in. And also, I think the reason why Julian suggested to use this, because again, I think in the paper, which is about the comparison of Panaru and all the other programs like Rory, they were able to use that set of options, which created a bigger core genome. So the main problem here, and this is what we already spoke about last time when we looked at those Staphylococcus psilosis isolates, is that you have to distinguish between two sets of genes. So let's say that you have 10 isolates to compare. You don't need a reference genome here. You just need the isolates. You need the POSQ files. You have to assemble them with any of the methods we already discussed. You have to annotate them. The easiest way to annotate is Proca. And then the only thing you need, as it's written here, or as it's written in this documentation, not this one, but sorry, I'm trying to go to the main documentation, get started here in this one. So you only need the GFF files. As you remember, when we did the Proca annotations, these folders for me. They are all the main outputs of Proca. So this is what you get back from a Proca annotation. You get back an FAA file where you have the protein sequences for all epileptic genes. You have a GBK file, which is the gene bank format annotation. You have the FNA, which is just a nucleotide FOSTA file. And you always have the GFF file. So this is the file that you have, which looks like this. So sorry again, the lines are broken at the end, but this is what you have, where you have an identification for the contig, and you have an identification for the gene, and then you have some kind of name for the gene. Of course, there are so many hypothetical proteins, but sometimes you will have a real product with a real function and annotation name. Also, that the gene name here will be C and not just a locus tag. So this is what you will get back, and it is coming up time to time that if you are using Proca in default settings, it doesn't have the best resolution for all your species, for all species one by one. So it is worth to make your own Proca databases. And if you think this is something we should discuss, I'm very happy to do this on Thursday. I think this would be very useful, actually, especially for those people who are working with only one species. If you are making a species or genus-specific Proca annotation database, then you will have much less hypothetical proteins and much more named genes and proteins at the end. So it will be much easier to see similarities or differences later on. But even if you are using Proca, or even if you are using Panaroo, you only need these GFF files. So one GFF file per isolate. And then again, you have to be prepared for usually quite a long run. I just tried to run Panaroo with on those 4, actually 5 GFF files, and it took a very long time. I don't know if Chioma, did you finish with your Rory run?
No, because it kept breaking and I didn't want to start it because we were going to have this class just in case we needed this.
Okay, so probably there is some problem when you are putting in 600. Okay, yeah, okay, we will check that. But yeah, it's as soon as you're going up, it will be way much more time consuming. According to the paper again, the Panaroo paper, this is also one of the advantages of Panaroo, that it's much faster. So I think we will have a discussion with Chioma after today's meeting. So probably I will be able to tell you a bit more about our experience with Panaroo on Thursday. So if you are running Rory, you will always get back a tree at the end. And this is what I had. So if you remember, we already saw the accessory binary genes f.fa.newick. Last time this was that tree that we visualized in Fandango. So this is a tree that is based on the gene set differences mainly. But there is also, so there are different things you are producing, either if you are using Rory or Panaroo. The 2 main categories you are generating is the core genome and the accessory genome. In Panaroo, let me just show you. This was the result of my Panaroo run. There is summary statistics. So the core genes means that 99 to 100% of the isolates or strains, different genomes you are using, are having all of these. So as you can see here, this is 936 genes that all of these are, this was the comparison of the 4 different Staphylococcus species that we discussed on Thursday. So the Xylosus, Equi, and 2 other. And then usually you have, okay, you have 3 categories. I mean, here you have 5, but as you can see, there are only genes in 3 of them. Usually there is a soft core, there is a shell. The soft core is also present in Rory and also the shell. So what you see here, of course, the percentages here are a bit misleading because we only have 5 strains in this comparison. So even if one is missing a gene, it cannot go into the soft core because then it will go down to 75%. No, 80%. But as you can see, there are quite a lot of genes in the shell, which means that these genes are shared between at least 2 isolates. So in our case, if one gene is present in 2 isolates, that's 40% of the total. So it is, of course, between 15 and 95%. And then we have we actually don't have any external accessory genes. That is a bit strange here. Total, or there is maybe an accessory genome file. This is what I haven't get the time to look yet, so forgive me. I will also. Oh no, I know, because if we have it only in one, that's already 20%. Okay, the reason why we have all the genes here, because if we have a gene only in one isolate, because it's 1 out of 5, that will be 20%. So it will be in the shell as well. So the whole result is much more informative if you are using, of course, more than 5 isolates, like 20 or 50 or 100, which you usually use. But the main thing you want to focus on, there are 2 main things. First of all, if you want to do a nice genomic distance tree between these isolates, and what we are speaking here now is not just the same species or the same strain. So for SNP, as I told you before, it is very good to use for very similar genomes. Here we are going one step up, so you can do core genome based comparison within species between strains. I think that's one of the best way how you can distinguish between strains or sequence types or clonal complexes. For that purpose, I think the core genome alignment is one of the best way to go. But also, I would go even up to genus level. So for example, what we did last time, we took 4 different species from the same genus. This is again what you can do. So of course, bacteria are not grouped together just by chance. They are grouped together because they have similar characteristics, similar phenotypes, or because they have similar genotypes, which means that they will share, of course, genes. And as soon as you have the core genome, you are aligning the core those genes to each other. Again, I don't think that there is any alignment done here yet with the Panaru. This is one I will check again. In Rory, if you finish the Rory run, then you can, you already have an alignment, I think. Let me just check. It is, the Rory has another site as well, where it is a bit more detailed how to use it. The Rory website. We will go through these next time as well. I hope I will be able to do some kind of so what I will do for Thursday is that I will show you how to run both Rory and both Panaru, sorry that I wasn't able to complete this task for today, but I just want to show you the main approaches, and then I will show you how to extract data from these runs. But also, yeah, this is what we almost had. And I think, yeah, so here what you do, there is already an alignment file, yes. So when you do the RORI, at the end, you will always have the core gene alignment ALN file. This is a multiple alignment file already. And from this, you will be able to plot a tree. And this is very important, that the tree that you will be able to do from the multiple alignment is not the same what you get back as the default tree with RORI. So in default, what you get back, this Newick tree, this is based on the gene presence absence. So again, you are defining the pairwise distance basically by looking at the shared genes between two species. So if you have 2000 genes and two different strains are sharing 500, then the distance between them, or the similarity between them, will be 25%, because they are sharing 25% of their genes with each other. Of course, this is completely different from a whole genome SNP approach, because we are only looking at the presence or absence of genes. At this moment, we don't look at the differences within the genes. We are recognizing genes by using some kind of gene annotation algorithm, and then we look for the similar genes, but we don't look deeply into the sequence of the genes, just the presence. If you want to look into the sequence of the genes, and this is what you can do with these further one step by using FAST-3, then you will have an alignment of all of those sequences that RORI extracted from the core genome. So again, it's very important that this is much more like the SNP kind of approach, but instead of looking at all the differences within the genome, you are only looking at differences within genes. And the most important thing here is that while having a whole genome difference, you can have SNPs anywhere. You can have SNPs in non-coding regions as well. As soon as you are looking at differences in core genes, genes that are highly conserved within a whole species or highly conserved within a whole genus, you will have much less variation, just because these genes are much more conserved. So if this is the other thing you always have to consider when you are choosing your approach. If you are choosing whole genome SNP differences by using SNP, you will have a very high resolution map, but probably you cannot go back in time too much. So you probably cannot go back like distance, phylogenetic distances. You can go back in a few 10 years, or maybe a few 100 years, maybe go back, I'm not sure. But it's much more relevant to recent changes. As soon as you are switching to core genome alignment, so first core genome extraction and then alignment, you can go back in time way much further, because simply you will have much less variation, and it will be much easier to track these variations from branch to branch. But it's also true that because you have much less variation in core genes and highly conserved genes, the core genome alignment is not as good for analyzing short-term changes. So I wouldn't, for example, I wouldn't use the Rory and core genome approach for a mutation accumulation study. There is a chance that there will be some mutation in the core genes, but I think it's much better to go with the SNP approach. And this is only one thing what you do, but the other thing is, and especially if you are looking different phenotypes, I will also try to make a dummy phenotype annotation file for Thursday, so we can see how you can group your isolates by some kind of phenotype. So let's say that you have 200 isolates and you are looking for antimicrobial resistance or growth on a specific media. So you will have that kind of phenotype information together with, of course, all the genomes, and you want to pair up these, and you want to check what is the change that made this phenotype. If you are losing something, if you are losing a phenotype, then there is a chance that you will have a mutation, again, a loss of function mutation in one of the core genes. But in many cases, there is a much higher chance that for some kind of phenotype, there is not a mutation that is creating that phenotype, but it's a new gene. Of course, we are within the prokaryotic world continuously, especially if you are thinking about antimicrobial resistance. That's probably the introduction of a new gene. So that's when you are going to the accessory genome information and try to extract those genes that are only, for example, present in one of these groups. Let me just show you quickly one example how you can compare multiple isolates. I will just take one of our old projects when we compared Staphylococcus chromogenes species to each other. And then you will see that sometimes you can have a nice big block of accessory genes that are only present in one branch of your tree. Right. So something like this. So as you can, I hope you can see something from my screen because it's quite small for me as well, but this is the tree here. And what you see here are, this is the same species, but different isolates. And as we found out later, these are all different, many different sequence types actually. Clearly there is a core genome. So this is what we call as a core genome. When you have the blue, so each one of these blue small lines here represents one gene. And as you can see here, if they are just above each other or overlapping, these genes are all present in all of the isolates. And then it comes to this point when a few of the isolates are missing that specific gene. But the more important thing is if that you have some very close related species. If you just, for example, look at this part here, I hope you can see this branch here. So this branch here are actually sharing these genes, which you can see here. So as you can see, these are all vertical lines, which means that for those genes, they are all present in this small group. Of course, there are bigger parts as well. So if you look at this, for example, this is the plot of 3 or 4 different isolates, and they are all having these genes. Of course, this one has as well. But usually this is what you are looking for. So if, for example, if you are finding some interesting phenotype in this branch, then you can try to find those genes that are only present or at least present in much higher frequency within this branch. I think it's also possible somehow to find, yeah. So if you just click on this branch, you can see here a kind of like histogram or graph about, so the blue line represents the frequency of each genes within the whole data set. So if it's up to 100%, that means that those genes, again, the genes are here at the top, one by one. So all of the genes up to this point are present in all of your isolates, and then it goes down, and the genes here are just presenting in one or very few isolates. But if you click on a specific branch like I did, and this is now blue, I hope you can see it's blue, it's very small, then you have an extra graph here with this purple. And the purple peak here means that within the selected branch, this gene actually represented 100%. And this is exactly the line of this one. And of course, you can zoom in like this. And if you are interested in what is this block here, as you can see, this block is only present in this one. So there is no other strains here. Then you can just simply go up and look at the genes. Of course, you will have again a lot of hypothetical proteins, but it's okay. But at least you will be able to say something about this. So again, you created this whole tree based on the gene presence and absence, not on the core genome alignment, and you can find out phenotype associated genotypes on this graph very easily. The same thing if you look at, for example, this one here, this branch, then you can see that there is a high peak here. It's always good to see those ones which are out of this core or shell genome, but have a high peak. So this is one of the ways how you can, for example, find a phenotype-specific genotype information. And finally, what I would like to speak one thing more today is what can you do if you want to compare multiple genuses, yeah. Or for example, if you have a metagenomic sample where you have a complete mixture of everything, but you want to do at least some kind of output about them. And this can be done by two different programs, but I think as far as I understand, they are doing the same. I'm using Mesh for that, but there are many people who are using Fast ANI, and that's what I also sent you as the link. So the word, the part ANI means it's just the average nucleotide identity, as you can see here. And the reason why this is fast, because it's actually doing the same thing as Mesh is doing. So as you may remember, Mesh is extracting small pieces from the genome. Again, it depends on you how high detail resolution you want to achieve. If you want to achieve higher resolution, you can extract more of these small pieces from the genome. We are speaking about those k-mers that are, I think in Fast ANI it's 16 nucleotides, in Mesh it's 21, but it's almost, there is no significant difference, I think, between these two. So let's say that you are extracting 1,000 of these small pieces from a genome, and you just simply count how many of these 1,000 can be found in species B, C, D, and E. So you are building up your own mesh database, and this is the same, again, what we did before, and I will also include this in this how-to or cheat sheet. And then you use this or screen function. I would use the screen function much more of the mesh, or use fastANI. As you can see, fastANI is also able to compare one-to-one, one-to-many, or many-to-many. This is what you need, many-to-many, when you want to end up with a distance tree, of course, within a big pack of genomes. Of course, you have to keep in mind that this is a very low resolution and very low reliability product what you are making with this kind of comparison. So if you really want to do something reflecting to I mean, if you want to do, for example, a time tree, or you want to do a real distance between two close related species, you would never use these kind of approaches. The k-mer-based approach, if we are speaking about fastANI or we are speaking about mesh, is only good for having a first picture of your results. Usually, a first picture of your many different species. I don't really see too much point to do mesh or fastANI, for example, in the case of Chioma's project, when she's looking for only Salmonella genus, or actually only a few species in Salmonella. The reason is that you will find a huge homology between those, and you don't really know if there is any real difference between 99.5 and 99.9% of identity, because it can be caused by not just by one or two SNPs, but also it can be caused by differences in the assembly. As you remember, if you have you may remember that if you have a fragmented genome at the end of your assembly, so your genome is not within one contig, but in multiple contigs, then if you are looking for a k-mer match that is actually on a boundary of two contigs, then you won't find it. So you are looking for a 21 nucleotide fragment, but 5 of this 21 nucleotide is at the end of contig A, and 16 nucleotide is in the beginning of contig B, then you won't find this. Even if the sequence is there, even if the sequence is completely matching, you won't find any alignment. So just by making exactly the same nucleotide sequence, exactly the same genome fragmented, you are losing identity percentage here, which is of course not really what I want to see. So it's not a real difference. It's just a difference coming from as a side product of your assembly process. So that this is why I think these kind of approaches are completely useless for comparing very close species to each other, but they are very good and very fast, and this is very important. They are very fast if you want to compare hundreds or sometimes even thousands of different genomes from very different sources, especially if you are thinking about, for example, metagenomics. This is a very good picture, first picture. Let me give you an example. When we did metagenomics in different samples from the same farm, so we sampled 10 different pig poo samples from the same farm, So you can predict that the pig's pig gut microbiota is quite synchronized between pigs and pigs if they are in the same farm, if they are, just swimming in each other's end products usually. So you can imagine that there will be a lot of very similar species between two metagenomic sequencing, but in the meantime, you know that there will be a huge amount of diversity within the sample. So if you choose a k-mer-based method like the FAST-ANI or the MESH, then very quickly you can find those metagenomics assemblies that are matching between two samples. And you can also see a kind of broad picture about the genus composition, for example, even if you don't have the exact match. So, for example, we had a lot of different lactobacillus species in these microbiomes. The same lactobacillus species, let's say Lactobacillus johnsonii, from two different samples came out from this kind of distance measurement with a very close distance. But we also saw a not much bigger a distance between Lactobacillus johnsonii and Lactobacillus reuteri. So between two different species from the same genus, you will see a lot of matches, not as much as between the two isolates from the same species, but you will have a broad picture about the whole organization of your sample. You can identify branches and you can identify genus level clusterizations within your sample. But that's all what you should do with this kind of approach, nothing else. You shouldn't go into details of genomes. You don't really want to do any timed tree from a comparison like this, but it is very good for, as I said, comparing a high diversity sample. So I think I will finish here because I already spoke too much. But again, the three main approaches you can choose is highly depending on your aims and your source. If you have, again, just let me just give you the last take-home message. If you have very close isolates from the same strain, from the same, probably the same source, but different time points or different patients, probably use a very high resolution comparison to find out the exact, the perfect, well-defined distance between them. So use the SNP, a whole genome SNP approach. If you are expecting a bit more distance, for example, because you are looking at the same species from different geographical locations or from different countries and different hospitals, for example, different hospital isolates, but from the same species, then it's very good to go up one level and do a core genome and define the distance by the core genome alignment, in the same time defining phenotypic differences by using the accessory genome. And if you want to find out if you want to do a quick picture of your high diversity sample, like for example metagenomics sequencing, then use a k-mer-based approach where you are extracting small pieces from each genome and try to find those small pieces in the other one. Okay, I will collect every information from all these websites for for Thursday. I won't go through of those. I mean, we won't be able to do it again because it's just too much time, but I really hope that you will have a chance to do it. I will also put up some test data set, so you can test your runs on that. But the most important thing, I hope you got that message, is that you always have to think forward what kind of method you want to use. So I will give you on Thursday this document, this help, but we will actually go and continue with the real creation of trees, how we can build up trees, what kind of different methods we can use to clusterize our samples, how we can visualize the tree, and also I think it's good if we go into a bit of visualization on like using ITOL and visualize phenotypes as well, not just the genotype differences. But I think quite a lot of you already used ITOL, for example, extensively. So, but I think it's nice to have a look on the ITOL as well. I will finish here, which means that we were very quick today. But if you have any questions, I'm very happy to discuss it.